Evening. We will be shortly starting with our course. Just before that, I would like to see we have the points. Okay, we'll start in a couple of minutes. Let's wait for everybody to join in and then we will start in a couple of minutes. Okay, good evening, everybody. Welcome to the weekly live interactive sessions for the course Enzyme Science and Technology, which is taken by Professor Vishal Trivedi of IIT Guwahati. I am Sanket from Kusuma School of Biological Sciences, IIT Delhi. And today is the sixth session out of the 12 sessions that we have, and we are halfway through the course. Today's session is about enzyme catalyzed reactions, biochemical reactions. So we will look at uh, the biochemical reactions that require enzymes. Almost all biochemical reactions in your body require enzymes. We will see how they work. Now, we can broadly classify the enzyme catalyzed reaction into two heads. One is the catabolism, and another one is anabolism. So, catabolism is where energy is produced, and anabolism is where uh, it's mainly where energy is spent for biosynthesis of molecules. We'll look at each and every one of them. So, for example, if we talk about catabolism, so carbohydrates, for example, it's converted to glucose that gives us energy through a pathway called glycolysis. We'll talk about it. Similarly, lipids are converted to fatty acids, which again gives energy. And then there is protein that is converted to amino acid, which again gives some energy. If we talk about biosynthesis of biomolecules, we have carbohydrates. So for example, when glycogen is converted to, sorry, glucose is converted to glycogen, that's a kind of anabolism. Similarly, for lipids, exact opposite, like when fatty acids or fat is there, glycerol is there, those kind of things. Then we have proteins, protein synthesis, and also nucleic acids that would include DNA, RNA, and so on. So talking about carbohydrate metabolism, so for example, glucose is converted to glycogen and it can go and get stored in the muscle or in the liver, right? Something like that. Glucose can go to the cytosol and undergo glycolysis, where it is converted to glucose 6 phosphate. Then through glycolysis, from 6 carbon, it is converted to pyruvic acid, which is 3 carbon, right? Then it enters the Krebs cycle, where it can produce ATP or NADH. NADH, it enters the electron transfer chain, and there it converts ADP to ATP. So these are all reactions which actually produce energy. Now, glycolysis is central to carbohydrate metabolism. Fine, and it's universal in both prokaryotes and eukaryotes. Fine. So a 6-membered carbon, that is glucose, is converted to 3-membered carbon, that is pyruvate. And then it enters the Krebs cycle, as you can see in the right? Enters the Krebs cycle. It's a 10-step reaction. And it requires 10 enzymes. So, for example, glucose is converted to glucose 6-phosphate. This is the first reaction of this. It uses hexokinase, as you can see. So here the enzyme is hexokinase. So basically, glycolysis can be divided into two, you know, um, I would say categories or two parts, where at the first it's the energy or two stages, the energy investment stage, and then it comes the energy harvesting stage. And there's a net gain of 2 ATPs in overall. But if we consider the ATPs that are getting from NADH, then we'll have more number of ATPs. But anyway, uh, the point is, so these are the enzymes which are used, like hexokinase. Then phosphohexose isomerase converted glucose 6-phosphate to fructose 6-phosphate. So the enzyme is phosphohexose isomerase. So let's see that. So first enzyme is hexokinase. Then we have phosphohexose isomerase. Then we have third enzyme, which converts fructose 6-phosphate to fructose 1,6-bisphosphate. The reaction is missing here. So fructose 6-phosphate to fructose The enzyme is phosphofructokinase. Then fructose, once this phosphate is converted into dihydroxyacetone phosphate. So there are two ways, either dihydroxyacetone phosphate or glyceraldehyde 3-phosphate. So this is done by the enzyme aldolase. There's the fourth enzyme, right? So this is a ketose and this is a aldose, right? And they can be interconverted using an enzyme called, again, this is the fifth enzyme, phosphotriose isomerase. In glyceraldehyde 3-phosphate that we have, these are both are 3-carbon sugars. So glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate in this reaction. So this is the sixth reaction, and the enzyme is glyceraldehyde 3-phosphate dehydrogenase. Seventh enzyme, 1,3-bisphosphoglycerate. It converts 1,3-bisphosphoglycerate to 3-phosphoglycerate by phosphoglycerate kinase. Then we get one enzyme, 3-phosphoglycerate is converted to 2-phosphoglycerate. Again, this is missing. So the eighth enzyme converts 3-phosphoglycerate to 2-phosphoglycerate. By the enzyme phosphoglycerate mutase. 2-phosphoglycerate is converted to phosphoenol pyruvate. This reaction, 9-step, using the enzyme enolase. And then phosphoenol pyruvate is converted to pyruvate using the enzyme pyruvate kinase. So at every step of glycolysis, we have one enzyme that is important in each step, right? Similarly, we have the citric acid cycle, where this we get pyruvate through glycolysis. So from glycolysis, if you remember, the end product was a pyruvate. Pyruvate, that is what we get in the end product. That is what is taken up by the citric acid cycle, right? And that happens in the mitochondria. As you can see in this figure, it happens in the mitochondria. And then following the citric acid cycle, we have something called the electron transport chain that would basically, generate ATP. It has the enzyme called ATP synthase, as it's marked in the slide, right? But that's not important. What is important right now is what are the reactions. So pyruvate gets converted to acetyl coenzyme A, which is enters into a cyclic process where first we get citrate, then cis aconitase. So citrate is we get by citrate synthase, right? Sorry, pyruvate dehydrogenase. Then we have citrate, and then we have aconitase, which gives you cis aconitase. The same enzyme then forms isocitrate. Then we have isocitrate dehydrogenase, which forms alpha ketoglutarate. Then alpha ketoglutarate dehydrogenase forms succinyl coenzyme A. Then this succinyl coenzyme A synthetase converts succinyl coenzyme A to succinate. And then this succinate is converted to fumarate using succinate dehydrogenase. And then it is converted to malate using fumarase, right? And then it is converted to oxaloacetate by using malate dehydrogenase. And then finally, we get oxaloacetate. So oxaloacetate gets converted to citrate using citrate synthase. And pyruvate carboxylase is the enzyme which is helpful for this insertion into the uh, citric acid cycle. Citric acid cycle is also known as the Krebs cycle or tricarboxylic acid cycle, also known as the TCA cycle. So if we look at the rate limiting step, so conversion of pyruvate to acetyl CoA, which is very important for uh, entry into the citric acid cycle. This is controlled by pyruvate dehydrogenase. And it is allosterically regulated basically inhibited by high levels of ATP, NADH, and acetyl CoA. When there are high levels of these energy, citric acid cycle is inhibited right in its very first step. Acetyl coenzyme A is not formed. Similarly, citrate synthase isocitrate dehydrogenase and alpha ketoglutarate, all three of them are inhibited by high levels of ATP, NADH, and succinyl coenzyme A. Similarly, isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. Both of these can be stimulated by calcium. Fine. So if you look at Krebs cycle once again, citric acid cycle, this is how it looks like. And it is centrally connected to all metabolic pathways, right? Uh, it's important. For example, and for if we take an example here, so for example, acetyl coenzyme A and citrate, these two are connected with lipid metabolism. And we will see that once we study about lipid metabolism. Similarly, alpha ketoglutarate, green one, and oxaloacetate, these two are connected with protein metabolism. And we will see that as well later. So, so the role in of PCA cycle basically, or citric acid cycle, or Krebs cycle, is essentially it is associated with running of electron transport chain, because it gives the necessary precursors for electron transport chain. Fine. And Krebs cycle has evolved organisms to adapt or adopt to high oxygen environment. Okay? So that's how it's important. Now, talking about the beta oxidation of fatty acid, it is basically sequential removal of two carbon fragments from, and at each of these steps, an acetyl coenzyme A is formed, right? And this happens as the bond between alpha and beta carbon atoms are broken. So we get acetyl CoA. So it happens in the mitochondria, and we will see how that happens. Right? Acids with chain length more than 14 carbon, they cannot cross the mitochondrial membrane, and for that we require something called the carnitine shuttle, and we will see how it works. That transfers this uh, long chain fatty acids into the mitochondrial membrane. And there are three enzymatic reactions to this fatty acid acylation, and then we will see the enzymatic reactions in specific. So first, what happens? So as I told, as we already told you, that so after every beta oxidation, there is production of acetyl coenzyme A. So let's say if we start with C16, it will give one acetyl coenzyme A, okay, and then it will form C14, and then it will give another acetyl coenzyme A. So this is 1, this is 2, this is 3, this is 4, 5, 6, 7, and then 8. So 8 acetyl coenzyme A in the long run. That is what we will get. So acylation reaction, as you can see, it takes from, so from the cytosol to the matrix using carnitine. So acyl coenzyme A will basically form, so fatty acids. So basically, let's say palmitoyl coenzyme A will form acyl coenzyme A using ATP in the process. And it's ATP dependent reaction. And the main reaction is acyl coenzyme A thiokinase. Or acyl CoA synthase. Okay, that is the reaction, and then it will be transported to mitochondrial matrix via the carnitine shuttle, because acyl carnitine will get inside, and it will get inside in the mitochondrial matrix, right? Where we will have the further beta oxidation process. So there is carnitine transferase 1, which is present in the outer membrane, CPT1. I don't know. Yeah, it's on the outer membrane. And then there is CPT2 that is present in the inner membrane. So here we have this enzyme performs transesterification. It forms acyl carnitine or carnitine translocase. So that will basically transfer the fatty acids. And then on the inner side, there is um, carnitine acyl transferase 2. Okay, it's in the matrix side of the inner membrane of mitochondria. So here it will transport back and it will convert it back to acyl coenzyme A, right? So if we talk about the stages of beta oxidation, so once the fatty acid acyl coenzyme A is transported to the mitochondrial membrane using carnitine um, using carnitine. So there is four process. One is oxidation. It's a cyclic process. As you can see, it's oxidation is the first process. So it is catalyzed by three isozymes of acyl coenzyme A dehydrogenase. Fine. And electrons extracted from the fatty acyl coenzyme A are transferred to FAD. As you can see, these are the electrons where we are transferred, and they give roughly 1.5 ATPs. Okay, and it goes into the respiratory chain, electron transport chain. Then we have hydrolysis, which is the second reaction. So trans A to enoyl coenzyme A. So H2O is added to trans coenzyme A, and it forms beta hydroxyl acyl coenzyme A. And the enzyme which is required for it is enoyl coenzyme A hydratase. That's why also known as hydration. It's very similar to fumarase enzyme. It's very similar to that. So it follows a similar pathway to that of the citric acid cycle. And this is very common even in case of urea cycle, you will see it has linked with interconnectedness with the citric acid cycle. The third part is oxidation, where beta hydroxyl acyl coenzyme A undergoes dehydrogenation. So this is the dehydrogenation reaction, and it forms beta ketoacyl coenzyme A via the enzyme. Here the enzyme is beta hydroxyacyl coenzyme A dehydrogenase. Fine. And here NAD plus, this acts as an electron acceptor. And it also generates some amount of ATPs there. And the last reaction is thiolysis, where beta ketoacyl coenzyme A is cleaved by the reaction with thiol group of coenzyme A to yield acetyl coenzyme A molecule and coenzyme A thyroester of fatty acid, okay, shortened by 2 carbon atoms, as you can see in the figure. So if you look at the oxidation of acetyl coenzyme A, let's say if we take palmitoyl coenzyme A, which is a 16 carbon fatty acid. So we have C16 at first, which will be converted to C14, C12, C10, C8, C. So after each and every cycle, it will be converted by. So in between all of this arrows, there is the beta oxidation cycle. And after each step, there will be acetyl coenzyme produced. An acetyl coenzyme A is a 2-carbon compound, so it's very easy. So this is 1, 2, 3, 4, 5, 6, 7, and 8. 8 acetyl coenzyme A are formed. And then it enters, all of this enters the Krebs cycle, as we already have studied. So it could be Krebs cycle or the TCA cycle, whatever you call it. Any of this cycle it will enter. Now, talking about the regulation of fatty acid biosynthesis and catabolism. So fatty acyl coenzyme A has two fates. Either get into the carnitine shuttle, right? And it gets oxidized in the final product. And it's a, as we already have seen, carnitine shuttle is a 3-step process. And it's an important point of regulation. We talk about regulation, this is an important point of regulation. Otherwise, it could be converted to triacyl glycerol and phospholipids. This is the second phase. As you can see in the reaction here, so acyl coenzyme A can finally form triacyl glycerol and phospholipids. And there are certain cytosolic enzymes for important for this. So if the fatty acids are transported to the mitochondrial matrix, then they are destined for beta oxidation. That is usually how it works. And malonyl coenzyme A regulates fatty acid oxidation. How? Malonyl coenzyme A inhibits acyl transferase 1, sorry, carnitine acyl transferase 1. And this is the very first reaction. As we already know, carnitine shuttle has 3 enzymes, 3 steps. So the first one, CPT1, is inhibited using malonyl coenzyme A. And this is also increased due to high glucose. So when there is high glucose in the liver, okay, so it can be increased. So high glucose will essentially prevent fatty acid metabolism, right? It can also be regulated using uh, a high concentration of NADH compared to NAD plus, right? So in this case, beta hydroxy coenzyme A dehydrogenase, the enzyme which is used here, this step is inhibited. High concentration of acetyl coenzyme A inhibits iolase, right? Vigorous muscle contraction, strenuous exercise or fasting, consumption of ATP is increased. So when there is, let's say, vigorous exercise muscle contraction, or let's say fasting, any of these things will increase the consumption of ATP. In that case, AMP will be uh, increased. So AMP activated kinase will basically get triggered, which will increase the fatty acid degradation because we need energy now. So fats are that's why burned when you fast or do something like that. So this will cause reduction in the malonyl coenzyme A levels. And as we know, malonyl coenzyme A, it can regulate this. So it will basically increase the activity. In turn, it will increase the activity of fatty acyl carnitine transferase. Insulin-dependent phosphatases, this is a specific enzyme, insulin-dependent phosphatase. It dephosphorylates ACC. ACC is acetyl coenzyme A carboxylase, right? And ACC is the enzyme responsible for synthesis of malonyl coenzyme A. Okay, and then it further inhibits carnitine acyl transferase 1. So we already know this. So this is how regulation happens in fatty acid biosynthesis, right? So talking about the amino acid biosynthesis, amino acids are formed using, if you see the central dogma, the DNA forms RNA. Now this RNA is recognized by ribosome, which further produces amino acids, right? So that is how it works. So if you can see, this is the mRNA here. Ribosome is there, which recognizes this RNA, and there is anti-tRNAs basically, which forms the anticodons, and they form these amino acids. These amino acids together are joined using the peptide bond, and they further form the protein, right? We know that. Well, among amino acids, it can be categorized. So amino acids can be categorized into three heads: the essential amino acid, non-essential, and conditionally essential. So like histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine, these are essential amino acids. They cannot be synthesized by the human body and needs to be obtained using a diet. So you need an external source. A plant-based diet or animal-based diet will replenish you with these amino acids. Non-essential essential amino acids like alanine, asparagine, aspartic acid, glutamic acid, hydroxyproline, taurine, and threonine. All these amino acids, where these three are basically modified amino acids. They are not a part of the 20 amino acids which are common. So they are non-essential because they can be produced by or synthesized from your body. So there is a pathway which can convert these amino acids into these amino acids. So they can be synthesized. Conditionally essential amino acids are essential during the times of illness, stress, or growth because during those times they cannot be synthesized. So arginine, cysteine, glutamine, tyrosine, glycine, ornithine, proline, and serine. Again, this is an unusual amino acid in this case. Fine. Now, talking about amino acids, they are principally derived from glycolysis or the citric acid cycle or the pentose phosphate pathway. Fine. So pathway intermediates, these derivatives provide the carbon skeleton for amino acids where amino group is provided either by glutamine or glutamate. So mostly that we will see. So this is, they provide the amino group. And the carbon group are from the pathway derivatives of these three pathways: glycolysis, citric acid, and pentose phosphate pathway. Right? Uh, some amino acids are synthesized by the organism, some are not synthesized. Based on that, we have the essential and non-essential amino acids, which we have already discussed. So in all amino acid biosynthesis pathway, we have the reductive amination of alpha keto acids like the alpha keto glutamate, right? And there could be transamination using transaminotransferase or pyridoxal 5-phosphate, any of these two. Now, if we look at the overview of amino acid biosynthesis, you can see, so glucose forms glucose 6-phosphate. This is the process of glycolysis. So glucose 6-phosphate can form ribose 5-phosphate, which can form histidine, right? Okay, so 3-phosphoglycerate, again a process in glycolysis, forms serine, glycine, and cysteine. Whereas, 3-phosphoglycerate can also form erythrose 4-phosphate, which is required along with phosphoenol pyruvate to form uh, tryptophan, phenylalanine, and tyrosine. These are also called the aromatic amino acids. We will see them later. But erythrose 4-phosphate we get through pentose phosphate pathway. The intermediate for that pathway. So as we already discussed, glycolysis or the pentose phosphate pathway or the citric acid cycle. Any of three of these will actually give you amino acids. So as you can see, two pathways are already covered, and here we have the citric acid cycle. Fine. So here you can see alpha ketoglutarate and oxaloacetate. These were the two intermediates. Alpha ketoglutarate can form glutamic acid, which can further form glutamine, proline, and arginine. And similarly, oxaloacetate can form aspartic acid, which can form asparagine, methionine, threonine, and lysine. So that covers all the amino acids. So if we look at the amino acid families, they can be 6 families. So 1, 2, 3, 4, 5, and 6. So the first family is the glutamate family. So the second family is the pyruvate family. The third family is the aspartate family. The fourth family is you can find out that it's very easy to figure out what family which amino acid belongs to. It's the serine family. Find serine. And the fifth family is the aromatic amino acid. So it's aromatic family. And the sixth family is histidine itself, because it's the histidine family has only histidine. That's very pretty clear. Glutamate family has obviously glutamate. It has glutamine. all the derivatives that it can form. Arginine and proline. Similarly, pyruvate family has valine, alanine, leucine, and isoleucine. Aspartate family has aspartate itself, aspartic acid, methionine, asparagine, threonine, and lysine. Similarly, serine family has serine, obviously, cysteine, and glycine. Similarly, aromatic amino acid, there is tryptophan, phenylalanine, and tyrosine. These are the three ones. So we'll see how that happens. So, for example, uh, if you look at oxaloacetate, which comes from the TCA cycle, it gives aspartate, which can form asparagine, methionine, threonine, and lysine. Threonine can further form isoleucine. Fine. So unlike thought leucine would give isoleucine, that's not the case. Similarly, pyruvate goes for transamination reaction, give alanine, valine, and leucine. Ribose 5-phosphate gives histidine. This is a separate family altogether. This is the histidine family. We already know that. And then there is phosphoenol pyruvate and erythrose 4-phosphate. So phosphoenol pyruvate comes from the glycolysis, and erythrose 4-phosphate from the pentose phosphate pathway gives us the aromatic amino acids. And we'll talk about it in detail because, in this case, um, we will talk about it in detail, basically. Alpha ketoglutarate, as we already know, this is a part of, again, TCA cycle, and it forms glutamate, which forms glutamine, proline, and arginine, right? Um, 3-phosphoglycerate, it forms serine, from cysteine and glycine. So in this case of arginine, it can be formed through glutamate. In mammals, even proline can form arginine. So there is some interchangeable reaction. So it can be formed using the enzyme called arginase. And basically, ornithine, which is a part of the urea cycle, we'll talk about the cycle. So ornithine can form arginine as well. That also can happen. In animals, usually glutamate forms arginine. It's a derivative. So again, uh, it's a derivative of on ornithine. For bacteria, there's a de novo pathway for ornithine synthesis. Talking about proline biosynthesis. Okay, if we talk about proline biosynthesis, so I talked about that in general. So it can be from ornithine, which or glutamine. Both can actually form proline. So there are two ways to go about it, and that's exactly the point that we were trying to tell you. So about aromatic amino acid, um, that we get using a very popular pathway, which is called shikimate pathway. So, and it is basically central to the production of all kinds of aromatic amino acids. So these two heads, I mean, these two has been discovered recently, but this before that, we only had this chorus mate, right? Uh, so there, this the first precursor that gives you the aromatic amino acid. So it forms prephanate, and then it can form tyrosine. So from here, there is a difference in pathways. It can form tyrosine or phenylalanine. Okay, and okay, this is how we are getting So in this process, in between, glutamate, in both the cases, in this and in this, both the case, glutamate is converted to alpha ketoglutarate using an enzyme called aminotransferase. So to talk about the regulation of amino acid biosynthesis, it can be regulated using by allosteric regulation. Again, something that you would really read in enzyme kinetics. What is allosteric regulation? And for example, glutamate synthetase is regulated by AMP, CTP, glycine, and alanine. The overall effect of inhibitors is additive. So if there are more than one inhibitors, so there's basically a feedback mechanism for this. So if there are more than one inhibitors, so there will be more inhibition. Hence, it's also called concerted inhibition. And sequential feedback inhibition is more pronounced in aromatic amino acid biosynthesis pathway or the shikimate pathway uh, in specific. So now talking about the nitrogen excretion and urea cycle, there are four key steps in the urea cycle. So formation of citrulline from ornithine. So as you can see, so ornithine, this is a wrong spelling, but ornithine to citrulline, that is the first step. And the enzyme is carbamoyl phosphate. Okay, carbamoyl phosphate synthase, that is the enzyme. So ornithine carbamoyl transferase converts carbamoyl phosphate ornithine together into citrulline. Arginosuccinate, this is the first step, that is here. Arginosuccinate forms via citrulline AMP intermediate, and here we have arginosuccinate synthase, the enzyme is 2. Then the third process, arginine formation from arginosuccinate using the arginosuccinate lyase enzyme, and here fumarate that is produced that enters the TCA cycle. And we will see this very soon. And then urea forms, and this reaction, so arginine can form urea and they can also form ornithine. And as we already said, that the reverse reaction ornithine to arginine is basically what we were talking about. So about amino acid synthesis. So there is an interconnectedness between the urea cycle and the TCA cycle. As you can see from this figure, arginosuccinate is common to both the cycle. This is the urea cycle, and this is the Krebs cycle or TCA cycle. So these two are interconnected with each other, right? And for amino acid degradation, there are again pathways where amino groups are first removed, and then the carbon skeleton is oxidized to compounds that enter the citric acid cycle, as you can see here, right? And there are several cofactors which are important, like tetrahydrofolate and S-adenosine methionine. These are important for this amino acid degradation. Now, to look at the amino acid degradation, we have three heads. So let's say alanine, cysteine, glycine, serine, even threonine and tryptophan, they form pyruvate, right? Uh, Isoleucine, leucine, threonine, and tryptophan, they form acetyl coenzyme A. These are the three main pathways. And leucine, lysine, phenylalanine, tryptophan, tyrosine, they form acetoacetyl um, coenzyme A. These are the three main forms of degradation. So let's now discuss about questions. So the step in hydrolysis, the step in glycolysis that results in the production of both NADH and ATP is conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate, or B, cleavage of fructose 1,6-bisphosphate to energy harvesting, okay, or energy harvesting of glyceraldehyde 3-phosphate, or water elimination from 2-phosphoglycerate. So energy harvesting step would be of in glyceraldehyde 3-phosphate. It results in production of both NADH and ATP. We will look at it. The high energy compound formed directly from dehydration of 2-phosphoglycerate, we have already talked about it, is ATP, NADH, FADH2, or phosphoenol pyruvate. So the answer is phosphoenol pyruvate. And if you see the glycolysis pathway here, the first question which talks about um, the energy harvesting step, which produces both NADH and ATP, that is here, conversion of glyceraldehyde 3-phosphate, energy harvesting step. And the ATP high energy compound at the end is pyruvate, dehydration of 2-phosphoglycerate. So this from here, if we remove water, we get this. So that's the point. In order to slow down the glycolysis when cellular energy levels are high, the phosphofructokinase are inhibited by AMP and fructose 2,6-bisphosphate, insulin and glucagon, ATP and citrate, fructose 1,6-bisphosphate. So the answer to this would be ATP and citrate, and we'll see how that works. And the step in Krebs cycle that involves the substrate level phosphorylation is actually conversion of alpha ketoglutarate to succinyl coenzyme A. So let's see that. If you see this figure, fructokinase 1 can be inhibited. These are inhibitors, citrate, bicytrate, and ATP, and it can be, increased using ADP and AMP. Similarly, in Krebs cycle, you would see that the conversion of alpha ketoglutarate to succinyl coenzyme A, that requires substrate level phosphorylation. The intermediate that connects lipid metabolism to Krebs cycle is malate, citrate, succinyl coenzyme A, or isocitrate. So the answer would be citrate. And the another one which is acetyl coenzyme A. Right? Apart from that, the activity of alpha keto glutarate dehydrogenase in Krebs cycle is inhibited by high levels of ATP, succinyl coenzyme A, Ca2 plus, or NADH. So basically, succinyl coenzyme A and NADH both can inhibit. The enzymes involved in breakdown of triglycerides during lipolysis is protease and amylase, hormone-sensitive lipase and lipoprotein lipase, amylase and pectinase, or catalase and cellulase. So any kind of lipolysis will involve uh, amylases and it will involve lipases. So lipolysis, that is breakdown of lipid, is usually happens through lipases. The final result of thiolysis reaction in beta oxidation catalyzed by acetyl coenzyme A acetyl transferase or thiolase, is a formation of a long chain fatty acyl coenzyme A, conversion of beta keto acyl coenzyme A to beta hydroxyl acyl coenzyme A, formation of water and CO2, production of acetyl coenzyme A, and shortened fatty acyl coenzyme A. So in case of beta oxidation, we have already seen that there is acetyl coenzyme A which is formed and a fatty acyl coenzyme A that is shortened by 2 carbons. So that is what we get. So this is the answer, option D. So you can see that thiolase will give you, uh, you know, 2 carbons less fatty acyl coenzyme A and the acetyl coenzyme A, as we were already talking about. Okay. Insulin influences fatty acid metabolism during high blood glucose level by inactivating the pKa, activating the AMP kinase, activates acetyl coenzyme A carboxylase, or inhibits acetyl coenzyme A carboxylase. So what would be the answer to this? I'll just give you a minute. We already discussed this today. So fatty acid metabolism is actually regulated by insulin, because insulin has insulin-dependent phosphatase. So insulin-dependent phosphatase dephosphorylates acetyl-CoA carboxylase, and which in turn activates it. And this is the reaction which is, this is the enzyme which is responsible for synthesis of malonyl coenzyme A. And malonyl coenzyme A is an inhibitor for um, fatty acid metabolism, or we can say beta oxidation in general, right? The step in fatty acid oxidation that is affected by the regulation of carnitine shuttle is entry of fatty acyl coenzyme A to mitochondria, synthesis of malonyl coenzyme A, activation of ACC, that is acetyl coenzyme A carboxylase, or inhibition of pKa. So carnitine shuttle, the work of carnitine shuttle we already know is because lipids which are, or fatty acids more than 14 carbon long, they cannot enter the mitochondrial membrane, and for this a carnitine acyl transferase is used to for entry of these into the mitochondrial matrix. So for that, entry will be prevented if we have uh, any effect in the carnitine shuttle. And you can see this in this diagram also. So carnitine transferase 1, as we already told you, carnitine transferase 1 is present on the outer membrane of the mitochondria. And then it's a three-step process. Basically, it will get attached to the fatty acid, and then it will form fatty acyl carnitine. Then fatty acyl carnitine will be transported into the inner membrane using a translocase enzyme. And then carnitine acyl transferase 2 will basically convert it into carnitine and the corresponding fatty acyl coenzyme A. The primary regulatory mechanism controlling the urea cycle's activity is feedback inhibition, transcriptional regulation, allosteric regulation of enzyme, or post-translational modification. So as we already know, urea cycle is controlled by allosteric regulation. We have already studied about it. The primary nitrogenous waste produced as byproducts of metabolic processes are actually it's ammonia and urea, as you can see in the figure on the right. So through this is in case of a fish, but whatever happens, basically all the nitrogenous base, they can be excreted either as ammonia or as urea. So ammonia cycle, urea cycle, and then there is ornithine cycle also in avians based on how much water is available. But these two are the major primary byproducts of metabolic processes. The coenzyme that is primarily utilized by aminotransferase during amino acid biosynthesis is ATP, NAD+, coenzyme A, or pyridoxal 5-phosphate. It's a very easy question to answer. It's pyridoxal 5-phosphate. The enzyme, I'm sorry. The enzyme that facilitates the reversible interconversion between alanine and pyruvate is alanine aminotransferase, glutamate dehydrogenase, aspartate transaminase, or pyruvate carboxylase. So in this case, as you can see in the reaction here, so alanine and pyruvate, they can be interconverted using the enzyme alanine aminotransferase, right? Um, so that would be the answer to this question. The pathway that serves as the central pathway for biosynthesis of aromatic amino acid, again, it has been already talked about pretty well in the lectures and in today's session as well. It's the shikimate pathway. So the shikimate pathway, which has this is the shikimate pathway, which has shikimate as the compound and chorismate as the precursor, which leads to all the aromatic amino acid, phenylalanine, tyrosine, tryptophan, And even naphthoquinone and aminobenzoate, these are not very common, but these are also produced using the same pathway. So shikimate pathway would be the answer to this question. With that, we come to an end to today's session. If there are any questions, feel free to ask. Any questions? Any questions? No questions? Okay, if there are no further questions, then we will be meeting again next week on the same time from 6 to 8 next Monday, and with week 7's content. So I think today we are done with today. So I'll upload these slides along with the recording as well, so you can follow it if you have not been following this. So I think that is all for today's session. If there are no further questions, I would like to stop the recording and then end this session.